Eta gaur, esan bezala, daukagu ekitaldi oso berezia, zeren gurekin daukagun pertsona da, pertsona oso ospetsua, harreman zuzena izan duena, Inigo San Millan doktorearekin, atletikoko esaten duten performance departamentuko buruarekin, eta baita ere Colorado Unibertsitateko irakaslea denarekin ere bai, eta eskerra berari, bera hemen daukagu, eta elkarte bion artean, Zientzia eta Teknologia Fakultatea eta EEUU guneren tartean, hitzaldi hau eta pertsona hau ekarri dugu, eta gainera, argita garbi, gure unibertsitatearekin egin duen lanagatik, omendu egingo dugu ere bai. So I'm saying that now we are going to have and it's an honor just to have with us a very important person from the University of Berkeley in California, that is going to talk us about the, maybe the one that it was announced, it was a little bit because I had announced the amazing story of lactate, key to understanding cellular metabolism in health and disease, and now it's the same, but a small change on that. And I'm going to tell that Dr. George Burks, Professor George Burks, is the world's leading authority on lactate and lactate metabolism. He has spent 53 years at the University of California at Berkeley, as I told you before, studying lactate, and thanks to his disruptive research, methods, and discoveries, we now know a great deal about the role of lactate in health and disease. Dr. or Professor Brooks' work has had and continues to have a major impact from the world of exercise physiology, that is the ones that we many times hear, but is going to the diseases such as cancer, type 2 diabetes, and Alzheimer's disease too. So it's a pleasure for us to have here George Bruce, Professor George Bruce. I'm going to give him the platform just to give us a small speech about his work and this important field he's researching in. Thank you very much.
Thank you very much. Ladies and gentlemen, it's a pleasure to be here today. Thank you for this kind invitation. Wonderful to see all of those of you here. So we made some new discoveries, so we changed the title a little bit. I hope that's okay. So time is short, so let's do the conclusion first. So there's glycolytic metabolism, and there's oxidative metabolism. The product of one is the substrate for the other, and the linkage is lactate. And this exchange or shuttling can happen within a cell. It can happen between organs and tissues. It happens at the whole body level. So we now know that lactate is a preferred fuel. We'll see actually over glucose. It's the major precursor to glucose, gluconeogenesis, and it is a signaling molecule. And we can't cover all of this today, but just some highlights of things. And it's a major route for body carbohydrate oxidation. And I'm particularly happy to see so many students and young people in the audience, also the older senior people, because this is going to be, in a way, some autobiographical and maybe an example for you to follow in your careers. So 50 years ago, everything was bad about lactate. It was an indicator of oxygen lack, a metabolic waste product, a fatigue agent, really indeed a poison. That was the idea. But where did these ideas come from? So I have a history of interest in this. I was a runner, and I show this to my students and even to my children, and they don't know who it is. But everybody was 18 years old. And this is in a place called Madison Square Garden, and it was the mile relay, or the equivalent of 4 by 400 meter relay. I tried very hard, but I couldn't reach the same level that I wanted to. So I asked my coach, why can't I make the Olympic team? And he said, "You have too much lactic acid. You have an oxygen debt." I said, "First I had an original sin, and now I have lactic acidosis, and I have an oxygen debt. What should I do?" And he said, "Go read about it." So I read about this man, Otto Meyerhoff, a famous man, well-deserved so in the history of biology and biochemistry. And he had this classic experiment. He had half a frog, and this frog doesn't have any oxygen. This frog muscle doesn't have any perfusion, but it has these electrical wires that made it contract. And he contracted it until the muscle stopped. Now, today we understand that in the absence of oxygen, glycolysis will go. But anyhow, when this muscle failed, it had a lot of lactate. So then the idea became that this accumulation of lactate is the cause of fatigue. It results in acidosis. It indicates an oxygen lack. And terms like the anaerobic threshold have been the result. So I guess for the athlete, it was no hope, so better go to the laboratory. Okay, and at the same time, there was another famous scientist, also in Germany, actually the professor of Meyerhoff. This was Otto Warburg, and he studied cancer cells, as indicated here. And if you know some German, this means a little oxygen, and this means plenty of oxygen. But the cancer cells are always making lactate when there's plenty of oxygen. And that turns out to be the case today. Because right now we're all making lactate, maybe me a little bit more than you, but we're making it nonetheless. So just to summarize, 50 years ago, or actually more than 50 years ago, a young man's dilemma, lactic acidosis, fatigue, cramps, the oxygen debt, and maybe cancer. Not good. But today we realize that lactate, as I indicated, is a fuel source. It's a gluconeogenic precursor. It's a signaling molecule. And our most recent discovery, we found that it is a major way to distribute carbohydrate carbon in the body. So not just during exercise, but after carbohydrate nutrition. So here's the traditional view of glycolysis. Glucose breaks down at the triphosphate level. We get two molecules, and then we have pyruvate formed, and the pyruvate is believed to go into the mitochondria for oxidation. And then traditionally, it's believed that if there's not enough oxygen or no oxygen, the pyruvate is diverted to lactate. I would submit this is wrong. So this is really the way we're looking at it right now. Most of it is actually correct. We have glycolysis leading to pyruvate, but most of it goes to lactate. So right now in us all, we have at least 10 times more lactate than pyruvate. And if we would run up the stairs, we would have 500 times more lactate than pyruvate. But that's no problem because we are adapted, our mitochondria are adapted to use lactate as a fuel. And we'll talk a little bit about that today as time allows. So here's the original idea of the lactate shuttle, and it was based on rodent research. So the idea is when we exercise, fast glycolytic fibers will break down glycogen to make lactate, and that lactate can enter the systemic circulation and go around the body and reperfuse the muscle bed. But some of the lactate can go to an adjacent red fiber, enter the mitochondria, and who would know because the only product was carbon dioxide. This was the original idea. We didn't think about that time about the brain. We didn't think about the liver. We didn't think about the signaling role of lactate. So we knew that transport was involved, but we didn't know about transporters. Now we know quite a bit about that. So just to summarize the key concept, there are some sites, tissues, organs sometimes, that are producers or driver cells, and then there are other organs and tissues which are consumers. And as physiology would have it, sometimes tissues can switch from production to consumption, like working muscle. But in this, mitochondria are really important because the mitochondrial network or reticulum is the site of disposal. So in biology, as in the physical world, things go from high to low. High pressure, low pressure. High concentration, low concentration. Mitochondria are important because they're at the bottom. They're the consumers of glycolysis, that is lactate. Okay, and this is a photograph from way back when. And my wife reminds me, Rosemary, Dr. Agostini, who's here, I used to have brown hair. But this is some time ago. But this is Casey Donovan, and this is a metabolism chamber. This is a treadmill that we had built for us. Here's a rat catheterized with a jugular catheter and a carotid artery catheter. So we could infuse tracers. We could study animals at rest, during exercise, before and after training. And we published several papers on this. But that was rats, and people tended not to believe us. So then we took this subject and we gave him some carbon-13 non-radioactive tracer, and we studied him, and then we got 5 more people, and we published a paper. So here are the results. There is lactate production all the time, here at rest, but about half of it is removed by CO2. So if you put in the carbon tracer, half of it's coming right back as CO2. And then, so there's continuous lactate production all the time. You're producing it right now. And, but you're oxidizing most of it. And then at 50% of VO2 max, you can see the production rate goes up and oxidative disposal goes up. So when we exercise, we actually oxidize more lactate than at rest. And then at 75% VO2 max, we have more production and also an increase in oxidation. So oxidative disposal is a predominant way. And it turns out the other percentage is gluconeogenesis. So a couple of take-home messages here. There's continuous lactate production. And lactate is produced because it's the obligatory product of glycolysis. But lactate is used, it's disposed of in ways that benefit us. Who knew? Without a tracer, you can't know this. And then during lactate production is high during exercise, especially simultaneous submaximal exercise. But again, lactate's removed, as I showed you, mostly by oxidation. Who knew? Without a tracer, nobody knew. So that was at the whole body level. But then we wanted to find out where in the body. So we took our subject. By the way, the subject's in this room. Okay, and this is a cardiac catheterization table. This is at University of California, San Francisco in the Department of Cardiology. Here is our cycle ergometer. We built it so we could study metabolism during rest and exercise. And over here on this side, I'm going to step over here. I hope I don't block anybody's view. We put in several lines. So first on the left arm here, there's an arterial sampling site. And over here, there are venous lines. One goes into the middle of the biceps. So the catheter would go from the wrist into the middle of the biceps. And this was going to be an inactive muscle. And we could sample blood from an inactive muscle. Another catheter went into the hepatic vein. So up the arm, down through the right atrium, down the inferior vena cava, into the liver, into the hepatic vein. And it's still another catheter that went to the iliac veins, which means up the arm, through the right atrium, down the inferior vena cava, into the leg, draining now the active muscle bed, simultaneously with the inactive muscle bed. And the last part, the one I'm going to talk about right now, is the coronary sinus. So those of you who have had anatomy know where the coronary sinus is. It's in the right atrium, right next to the AV node. This is the venous drainage from the myocardium. So now we're trying to get the arterial venous difference across the heart muscle. And it's right next to the AV node. So when we put the catheters in, you could also see some interesting waveforms because bumping the AV node causes that. So then just to summarize this, we see that during rest, there's a lot of dependence on fatty acids. This was not new, this was known, but it's important to validate that our results are consistent with the literature. And then 4% glucose, and even though lactate is low, less than 1 millimolar, it's 13%. Now during exercise, and here only 40% of VO2 max, we can see lactate now is the major fuel. And fatty acid oxidation increases absolutely, but percent-wise its role is reduced, and glucose is 4%. So this is an example of lactate shuttling. The muscles are fueling the heart. And without the tracers and without the catheters, nobody could know that. So a couple of take-home messages. During exercise, blood lactate from working muscles and other sites fuel the heart. Who knew that the muscles were fueling the heart? Well, unless you made the measurements with tracers and AV difference, you couldn't know. Now, in terms of translation to medicine, think about cardioplegia, or treating a heart and heart failure, or a person after a myocardial infarction. The idea emerges we could give lactate to fuel the injured organ. So going on from there, we wanted to know what's the effect of training. So we had some whole body data, some organ-specific data, and so we did another study. We studied men before and after training, and we used pulmonary gas exchange, but we had pulmonary gas exchange also to collect the carbon-13 CO2 from a tracer lactate. We had femoral, arterial, and venous differences, and so these catheters are inserted in the inguinal area and go down into the working muscle. So for this, you really need to train a skilled cardiologist who is doing cardiac catheterization all the time. We used a combination of isotope tracers, carbon-13 lactate. This is, those of you know that most of our carbon is carbon-12. Carbon-13 is mass plus 1. It's a little heavier. But in mass spectrometry, you can see this. And also for glucose, simultaneously, we used dideuteroglucose, D2 glucose. And deuterium is mass plus 1 over endogenous glucose, but if you have dideutero, it's mass plus 2. And we took biopsies and we did supervised training, and we studied people after training. Okay, so before and after training. And here's a movie of what was going on. So you can see the catheters. They're the white tubes that are in the femoral artery and the femoral vein. Dr. Wolfel here was chief of cardiology at the University of Colorado. He's sampling on the venous side, and Dr. Ann Friedlander is sampling on the other side. So again, before and after training, easy exercise, hard exercise, same relative and absolute power outputs. Okay, to summarize the results, if you just look at the top line, that really encapsulates a lot of things. So here's a picture of Dr. Wolfel and Dr. Friedlander, and this was Brian Bergman's doctoral dissertation. He's now on the faculty at the University of Colorado. Dr. Wolfel is retired. A lot of my colleagues and students are retired. What's the matter with me? But if we look at here the clearance, the oxidation of lactate in the muscle, so we know that a certain amount of labeled lactate goes into the muscle, and 100% is coming out on the venous side as carbon dioxide. So we can see here that what's happening is that training increases the ability of muscles to consume and oxidize lactate. And of course, this is going to be traceable to the mitochondrial network because that's where oxidation takes place. So training increases the mitochondrial mass, and that allows us to have a greater lactate clearance. So another couple of punchlines. During exercise, the working muscle simultaneously produces, consumes, and oxidizes lactate. And training really improves lactate oxidation. Who knew? Well, without the AV difference in the tracers, nobody knew. Now, a little bit of use of isotopes to inform about what's going on. So we give 13C lactate, and we can look at its exchange across tissues. That's true. And we can collect the expired air, and the 13C from lactate, when it gets oxidized, comes out in the breath, so we know oxidation, and also oxidation in the working muscles that I just described. But this carbon-13 is mass plus 1. So this tells us the amount of glucose that came from lactate. This is during exercise. And of course, this is the role of the liver and kidneys. So here are the data. So there's gluconeogenesis in post-absorptive men. These are all men, before and after training. And the training is here. So you can see that training increases gluconeogenesis at rest. So trained people have a real advantage in terms of maintaining blood glucose because they're training not only their muscles and their heart, but also their liver. So there's a big increase in gluconeogenesis. You can see this even at rest. And then over here is 45% of VO2 max before training. You can see gluconeogenesis goes up a bit. But then at 65%, gluconeogenesis goes down before training. This is probably because of shunting of blood flow away from the liver and kidneys to the working muscles. But then after training, this is the same power output. Same power output. You can see the big increase in gluconeogenesis after training. And this is over here on the right-hand side is 65% of VO2 max after training. But again, the power output has gone up about 40%. So there's a lot more exercise. But still, you can see training improves gluconeogenesis. This represents about 25% of the lactate disposal. That is the conversion to glucose. So training increases gluconeogenesis during rest and exercise. So training, and so on the top right-hand corner are two Nobel laureates, Gertrude and Carl Corey, who are known for the Corey cycle. So training improves the ability to maintain blood sugar level, use glycemia by increasing the capacity for gluconeogenesis from lactate. Who knew? Well, not even actually the Coreys, because the Coreys studied dogs, not people. They never studied people. They didn't study exercise. They didn't study exercise training. And they didn't study people before and after exercise training. So not even they knew. So we invented lactate clamp technology analogous to glucose clamp technology. So it could ask several questions. Well, if lactate is good for you, can you eat it? Can you drink it? Well, when you eat carbohydrates, you're making it. That's true. You can put it in a sports supplement. That's true. Some foods contain lactate. That's true. You can drink it. We put it in a sports drink. It worked really well. You can infuse it into a vein, and you can use it for treatment of injuries and illnesses, as I already suggested with cardioplegia. And the answer is yes. And we don't have time to go into a lot of it, but this was the dissertation work of Dr. Ben Miller, who's now at the University of Oklahoma. That's him in the top left-hand side. So we know lactate is the preferred fuel over glucose. So with our tracers, we can measure glucose and lactate metabolism simultaneously. And when we give lactate, it suppresses glucose uptake. But even more important than that, lactate infusion raises the pH. So it's really an alkalotic effect. Remember from chemistry, the salt of an acid is a base. So if we're giving sodium lactate, it's actually a base. So it helps to manage acidosis. So in terms of translation, think about it. Not only is it cardioplegic, we can use this substance to manage acidosis. We worked with UCLA Neurosurgery, and this is a positron emission tomograph picture. And this relies on an isotope of glucose that can be visualized. So if we look on the left-hand side over here, we can see somebody who has a brain injury. This is somebody who was in an automobile accident. So where red is, that's where glucose goes. And here on the right side, you can see glucose coming in the prefrontal cortex, but they have a prefrontal cortex injury right here, no glucose uptake. So the injured brain is starving for glucose. The next day, we infused lactate to 4 millimolar. Those of you who are sports oriented, you know 4 millimolar is sometimes thought to be an important level of blood lactate, 4 millimolar. You can see lactate completely substitutes for glucose. So for the brain, lactate is a preferred fuel. So think about it. Think about giving lactate to treat traumatic brain injury, as in this case. Okay, we're in the home stretch now. Everybody with me? Anybody want to get up and stretch? Move your mitochondria? Okay, so I talked about mitochondria before. They're important, not just biologically, they're important as the site of lactate disposal. And they're sort of the basement, they're the sink where things go. Here's an electron micrograph from Hans Hoppler. I show this because he studied human muscle. But you can see the mitochondrial network. So people think that mitochondria are these little jelly beans that float around in our cells. Not true. When you take a picture through the muscle, if you take it in the right plane, you'll see this network. So we actually helped discover this network called the mitochondrial reticulum. We call it the cellular energy highway. Others call it the cellular power grid. So this is where oxidative phosphorylation and most ATP is made. And most recently, we've looked at this with a three-dimensional microscopy. So I was talking to Professor Samalan about when we were kids with our crayons coloring. So if we had green and we had red and we mixed them together, we got a yellow color. So the same thing happens in immunocytochemistry. So if we have an antibody to bind to the lactate transporter, it turns green. This is a mouse muscle, and look at the abundance of the green around the perimeter. This is MCT1, the lactate transporter. The muscle's loaded with this. Now, also, we can do co-localization. So we can have another antibody. We can make it fluoresce red. Remember, red and green give us what color? Gold. If you're an athlete, what do you want to go for? Gold. So where you see gold now is where the lactate transporter and the mitochondrial pyruvate transporter exist simultaneously. This is made from the serial sections, many, many sections, 100 represented here, just a small distance within the muscle of this mouse. But you can see the abundance of lactate transporters that are not on the cell surface just, but also in the mitochondrial reticulum or the mitochondrial network. So the picture of mitochondria is completely different. The understanding of its functioning is completely different. So some years ago, we were asked by the Journal of Physiology to write a review paper, and we used for our symbol of this paper, the phoenix rising from the flames. So the old idea was that lactate caused burn, cramps, the oxygen debt, the anaerobic threshold. But now we're emerging from that. We realize that lactate's involved in cell signaling. It has a number of metabolic advantages. It's a major fuel. So like the phoenix, that's now undergoing in the scientific literature a changing of thought. And the phoenix was used to symbolize that. So in the paper, we had two depictions of lactate shuttling. So here during exercise, and down here is the muscle and skin producing lactate, and the liver and the heart, and so on. But also had the forethought to think about what happens in the gut, because exercise is a time when there's carbohydrate carbon flow. Glycogen, glucose break down, make lactate. That's an exercise. But when else in our daily lives is there carbon flow? Well, after we eat carbohydrate. That's another thing we thought about. So over here in this picture, we have indicated the role of the gut. I think they're cutting me off because the battery's going dead. Okay. Can we see the cursor? No, we can't. Okay, and so there was some data in the literature based on dogs and rats about the role of the gut in lactate metabolism, but there were no data on humans, so we decided to do that. So again, this is a photograph from one of our studies. So what we have here is we have the pumps containing the tracers, which are glucose, lactate, and glycerol. Glycerol is a marker of fat metabolism, of fat breakdown. That's on the venous side, and also on the arterial side, over here, we're taking measurements of glucose, lactate, glycerol, fatty acids, concentrations and isotopic enrichments, and we're also measuring insulin and counter-regulatory hormones. And finally, for this picture, we're collecting expired air, because again, if we're putting in a carbon-13 tracer on lactate and it's burned, comes out in the breath as CO2. And that's, well, I already told you that. At rest, it's about 50%. In exercise, it's close to 100%. Okay, a couple of slides here to illustrate this. This was recently published in a well-known journal. So here are our subjects. They are studied here at 7 a.m. So the lab folks we had to get there at 4:30 to set this up. We had to start the isotopic infusions. The subject came in about 5 o'clock. We did a background isotopic infusion, and then the person took an oral glucose tolerance test exactly at 7 a.m. So here's lactate, and these are post-absorptive people, so lactate is less than 1 millimolar, and over here, over 90 minutes, the blood lactate is falling. So we took these two values here as an average as the pre-oral glucose tolerance test, at which time now, at 7 a.m., the person drank 75 grams of glucose. This is a standard test, oral glucose tolerance test. So if we look here at the results, we can see that there's a significant rise in arterial lactate. Now again, ladies and gentlemen, I said we gave glucose. The first thing to rise in the blood is lactate. And the lactate reaches a peak at about 15 minutes, and then it falls, and then there's a secondary rise. And we'll try to explain why this is happening. So we're thinking that the enterocytes, cells lining the gut, are taking glucose and making lactate. And that's why the first thing to rise in the blood is not glucose, but lactate. And then the liver is holding most of the glucose and starting to release it after about a half hour. And then the muscles and other parts of the body are taking glucose and making lactate. So we have two phases of this lactate story. We have the enteric phase and we have a systemic phase. So there are two phases. And again, I need to show you the glucose phase. So glucose is low and stable before people drink the sugar. It doesn't respond in the first couple of minutes, but after about 15 minutes, the glucose starts rising. This is the standard kind of waveform here. So had we not taken samples at 5 minutes, and had we not taken arterial samples, we would have missed this whole story. But we thought about it in advance. So here's now accounting for 75 grams of glucose. About 13% goes to lactate. About 11% goes to glucose from lactate. Now, so together then, it looks like half the glucose that was taken in the drink goes to lactate, directly or indirectly by gluconeogenesis. So now we can start to summarize here. So we have lactate shuttling, and we know it's involved in cell fueling. We know it's involved in gluconeogenesis. I didn't talk much about cell signaling, but lactate crosses the blood-brain barrier and goes to our areas of appetite and suppresses our appetite. So I'm sure some of you in here have run 400 meters all out, or maybe 1,500 meters all out. Were you hungry when you got through? No, you were not. If you weren't throwing up, you were not hungry for hours. This is because the lactate is suppressing appetite. So it's actually doing some exercise is an appetite suppressant. And we know now also from our collaborations with clinical endocrinology at UC San Francisco, it's involved in reproduction. The development of the blastula is really important on the lactate level. And now in green at the bottom, lactate is involved in dietary carbon flux. So just not during exercise, but whenever there's a carbon flux, lactate's going to be involved. Exercise or dietary carbohydrate. Thank you for your attention. This is a photograph taken the day Dr. Agostini was studied. I was the first subject. She, my physician wife, was our second subject. Here are some of our undergraduates who work in the lab and some graduate students. And we had even the local collegiate champion, shot put champion, was an undergraduate working in the lab. So here's where we started. Here's our conclusion. There's glycolytic metabolism, there's oxidative metabolism. The link between the two is lactate. Lactate's a fuel, a gluconeogenic precursor, a signaling molecule, and a major route for dietary carbon flux. So at a more basic level, it's just not exercise, just not nutrition. It's about carbon flux in the body. Thank you very much for your attention. Thank you, Pat. Great. Thank you. Just going to go quick.
Hello, everyone. Thank you very much, and eskerrik asko for being here. And it's been a great pleasure to have Dr. George Brooks over here. My name is Íñigo, and I'm here. I used to be a student here at the University of the Basque Country, and where I did my doctorate here in one of these buildings here with doctors Johnny Dazusta and Javier Gil, who I believe they're here. And they were great to me because they were really hard on me, and I wanted to kill them more than once, but I finally was able to break through and finish my doctorate thanks to them, so I'm always really appreciated. And I did my doctorate thesis based on the work from Dr. George Brooks at a very, very small scale, but yeah, it's just that's when I got very involved into lactate. So, but I want to really emphasize on the work that Dr. George Brooks has been doing for 53 years. As we have seen pictures of since the 1980s, he was already making breakthrough discoveries. And he was the subject of those ones. So back in the days, because several Nobel Prizes that were saying that lactate was a waste product or the result of anaerobic metabolism, not many people believed him. But he never gave up, and he was swimming upstream all these years. And finally, now that we're finally understanding cellular metabolism and bioenergetics, his name now is very well known around the world, not just in the area of physiology or exercise physiology, but in multiple diseases. And I just want to I have 5 minutes only, I promise, but I just want to do a short trip of about 100 years long summarized at a very high level about the understanding of human bioenergetics and how someone like George Brooks has been one of the key players for us to understanding. It's been a big effort of about 100 years for multiple talented and hardworking scientists around the world, the improvements in technology to get us to a great moment in science today, where we are at a special moment to understand health and disease better at a level that we have never been able to understand. We spend a lot of time with genomics, which are very important, but there's a renaissance of human cellular biology and metabolism, and George Brooks is one of the most important people for this. So all this trip to understand bioenergetics has allowed us to really complete the picture, and now it's a great time because we start to do reverse engineering, and it's allowing us to understand health and disease better, and therefore opening new doors for diagnosis and therapeutics in different diseases like cardiovascular disease, type 2 diabetes, cancer, Alzheimer's. So these are going to be fascinating years ahead based on the knowledge that we have about cellular metabolism. So real quick, everything starts by Albert Einstein. You may think, how in the world Albert Einstein could be part of the human biology and bioenergetics? Well, it's very easy. Einstein was obviously in Germany, was very good friends with Otto Warburg. Otto Warburg's dad. He was Emil Warburg, was a very prominent chemist, and they were part of the High Society of Berlin in the 1920s. And then, Otto Warburg decided to go to the First World War. And then he just was not understood. People didn't pay much attention to what he was saying or trying to show to the world as a scientist. So he left everything and he enlisted to go to war. So his mom was very worried about him. He wanted him to come back to the laboratory and not to die in the war, but he would not listen. And then they were very good friends with Albert Einstein, and they asked Albert, hey, Albert, could you please write Otto Warburg and convince him to come back to the laboratory and leave the war field? So he wrote Warburg. I forgot to put the letter because there's a letter that exists. And he convinced Warburg, and Warburg came back to the laboratory. And then when he came back to the laboratory, that was a very important moment in history to understand cellular bioenergetics, because then Warburg started to unveil his magic and his science. He started to work with multiple experts like Otto Meyerhoff, who came to his lab to continue learning, even if he had already a Nobel Prize. But they were trying to understand glycolysis, as we know. And it was back then, it was called the Emden-Meyerhoff pathway. But already Meyerhoff was a student of Warburg. Now we know that Severo Ochoa was a student of Meyerhoff and also was a student of Warburg. He's the father of modern neuroscience. So another Nobel Prize that we have, and because of him, we know a lot about neuroscience. Then they started to complete Meyerhoff with Emden, the glycolysis pathway. And then we needed to understand what the hell happened once pyruvate or lactate enters the cell. And that was the Krebs cycle that we all know and we have studied. So guess who discovered the Krebs cycle? Hans Krebs, which was another student of Warburg, right? So all these Nobel Prizes and amazing minds, they had one person in common, which was Otto Warburg. And nothing would have happened if Einstein wouldn't have sent a letter to the war, right? Now, all these brilliant minds, they encounter one thing, which is the end of everything they were looking at, they were studying, and that thing was lactate. And back in the day, they said, this is waste product, this is anaerobic metabolism. So we kind of forget about this is poison. Until Dr. George Brooks came along and swimming upstream, he proved them all that they were wrong, thinking that lactate was a waste product. So this is one of the reasons why, because now we can take this to multiple fields in science. So a lot of people are talking about a possibility of a Nobel Prize down the road for Dr. Brooks. And that was one of the key pieces of the puzzle to complete about cellular metabolism. Because now that we have this, I'm not sure to say the final piece, but it's the end of the road of glucose, is the end product, as he has been able to prove over the years. Now we're in a position to really understand cellular metabolism really well, so that we can, through reverse engineering and understanding the processes of this cellular metabolism better, to understand disease better. Most diseases, if not all, have disruption or dysregulation of cellular bioenergetics, whether it's at the mitochondria or the pyruvate or lactate, but there's disruption of cellular bioenergetics. Once we have now the entire picture, we're in an amazing position to be able to understand disease, and therefore be able to come up with multiple diagnoses and therapeutics. So this is one of the area where, with the knowledge that I've been acquired working and having George as a mentor, we've been together embarking in an area of trying to understand the role of dysregulated lactate in multiple diseases. And I'm not going to go through anything, but we have several publications, and we continue trying to do more in cancer. We have one about the dysregulated mechanisms in mitochondria, but we have, especially in cancer, we're showing that it's a major player in cancer, in carcinogenesis. And then we have other studies that hopefully we'll get in different diseases. But anyways, I just wanted to summarize all this about an amazing journey of over 100 years that got us to today, and puts us in an amazing, beautiful situation to understand cellular metabolism and be able to cure diseases that are not being able to have a cure until now. And Dr. George Brooks has been one of the key elements to help us solving the puzzle. So thank you very much. Thank you very much, Dr. George Brooks. And we're open for questions. We have about 5 minutes or so of questions. Yeah. So questions? Anybody? Is there a microphone?
Good talk. Thank you. I have a microphone.
You have a microphone.
Yeah, thank you. That was a great talk, Dr. Brooks. Thank you so much for coming over and teach us such a wonderful conference. Now, my question is concerning the lactate signaling abilities. What kind of receptors can lactate bind to?
Oh, so that's a really good question. How does lactate signal? By changing, first of all, redox, the NAD-NADH ratio, which is a big regulator of metabolism. Also, I took the slides out in the interest of time. Lactate actually binds to a receptor. It was an orphan G protein called GPR81. Now it's called HCAR1, hydroxycarboxy acid protein 1, which works through Krebs, and the function of that is to inhibit lipolysis. So there are multiple ways that lactate works. It works by allosteric binding to receptors. It changes redox, and most recently it's been found that it affects lactylation of histones. So lactate labels proteins, and it labels histones, and it affects the unraveling of the histones for the expression of genes. So lactate is involved in a powerful way in metabolic signaling. And of course, one of the big things we know for training, we can double the mitochondrial mass. So lactate is probably involved in mitochondrial biogenesis by a combination of these effects.
Thank you.
Thank you for the question.
Thank you.
There's a person here. Aquí hay una persona. Ahí.
Could you just talk a little bit about the relation between lactate and cancer via the Warburg effect, please?
So the question, as I understand it, is how does lactate involved in cancer?
The relationship with the Warburg
Yeah.
effect in cancer. Yeah.
And Inigo is actually better equipped to talk to that.
Well, one of the things that we have seen and we have shown that lactate acts as an oncometabolite. And as Dr. Brooks said, it labels the histones and opens them up and allows lactate to express those genes, right? What we have shown in breast cancer cells, that lactate expresses those genes. Well, it's a major player in carcinogenesis, and we have multiple studies now coming out on other different types of cancer as well. And we've been able to, in one study that has been just recently accepted, we have been able to block lactate production, and we've been able to knock out the expression of a major component in cancer, which is EGFR, which is a major signaling pathway for growth and proliferation.
Thank you.
Yeah, so I hope that was an answer by the discoverer of the effect of blocking lactate dehydrogenase. And you know, just think about it. The Warburg effect is 100 years old, and so this signal, this lactate signal, has been with us for 100 years. And then leave it to somebody and say, well, let's block the enzyme that makes lactate. And he's been able to take cancer cells and make them normal by blocking LDH.
Otra pregunta aquí.
Hello. Thank you very much for the great talk. I was curious, so you mentioned that lactate actually has a protective role in case of heart and brain injuries. So I was wondering if this is completely attributed to its function as an energy-rich substrate or due to different signaling effects.
Yeah.
How is this described?
Yeah, so our thinking originally was that it was its preferred substrate. But now with the further development, evolution of this, it is undoubtedly a substrate. So we can actually increase, with isolated hearts, giving lactate increases cardiac function. So we know that. But we really suspect also that the lactate is involved in signaling and recovery and repair. So think about it, this injury, it occurs acutely, and so lactate is a good substrate for then, but then we need to recover and repair. And it's probably involved in both phases of the problem with the heart failure and myocardial infarction.
Tenemos para esto.
Congratulations, beautiful work. So just a very naive question. Do you think that during exercise also lactate fuels the brain?
Does lactate enter the brain during exercise?
Fuel.
Does it fuel? Yes, the answer is yes. So one of our former postdocs, Takeshi Hashimoto, went to Copenhagen and worked with Niels Secker. And they put in jugular bulb and arterial catheters. And they could see during exercise, lactate rises in the blood, goes to the brain, and fuels the brain. And this is an important result for physical educators working with children, okay? So what is recess? The kids run around, they raise their lactate, and one of the things about this study, they gave these subjects psychological tests, the computer test, memory tests, and learning tests. So they exercised, they raised their lactate, they got smarter. Just think about how kids play on the playground makes them smarter, more receptive to the lesson afterwards. And then they also not only did that study, they did a follow-up study. So they went the lactate rose, and they got smarter, and the lactate fell and went back to baseline.
Thank you.
Maybe teachers will have lactate pills. Yeah, you're not doing so well, have a lactate.
Well, right, maybe last question. Okay.
Thank both of you for your talk. It has been really, really nice. And I have a question regarding your Olympic teams idea when you were younger. And I was thinking that mainly thanks to you, we know the important role of lactate in our physiology, and that there is like a highly clear equilibrium in these bias. So nowadays we know that microbiota can help degrading lactate, like for example Legionella species, something like that. So now my question is, what do you think about this, that your coach told you that you couldn't go to the Olympics team because of your lactate levels? What do you think nowadays about that?
Yeah, so I think I hopefully impressed on you the important thing is clearance, the removal. So by training, you increase the mitochondrial biogenesis and the transporter expression are all around the body. So now you're better equipped for disposal of lactate. And I think that's an important role, but you also mentioned the microbiota. And this, we need to understand this better. We know the gut is lined with lactate transporters. And so the gut actually can produce lactate. And the gut can consume lactate. So, and the transporters are concentration dependent. So when you exercise, well, we're clearing lactate, right? We're burning it, the heart's using it. Probably some of it's going into the gut. So we actually want to study this. And I've got some ideas about how to do that, to take the tracers and encapsulate them. So some will enter the circulation from digestion in the upper gut and then also in the lower bowel. So I have some ideas about how to get things to the lower bowel and look at the role of the lower bowel in terms of lactate kinetics. But thank you for the question. It's really insightful because a lot of science now is about the microbiota in health and disease. And so we know if you exercise, you have a healthier microbiota, but you're also training the intestinal wall.
Okay. Thank you very much, eskerrik asko.
Bueno, hoy es un día muy especial porque hoy es el día en el que comienza la ciencia STEA y de hecho la rectora de la universidad está en estos momentos haciendo el acto de inauguración de ciencia STEA. Pero como reconocimiento pensábamos que podía ser una cosa muy interesante darle como uno de los obsequios sería esta camiseta de ciencia STEA que hoy en día es muy representativa de la propia universidad. Gracias. Y asimismo, como esta jornada está relacionada entre EEUU y la propia Facultad de Ciencia y Tecnología, y teniendo en cuenta del trabajo continuado que ha tenido el profesor George Brooks, pues la propia universidad quiere hacerle un pequeño obsequio y para ello, en representación de la misma, tenemos al secretario general y asimismo también al decano de la Facultad de Educación y Deporte, Julio Calleja. Por favor. Very good.
Professor Brooks, a couple of years we had a lunch in Denver during the ACSN conference with Íñigo, your wife, and your team lab members. 2 years later, I really appreciate it to be back, and for me it's a big pleasure to be here. In the name of the University of the Basque Country, we want to recognize your contribution in the field of sports physiology and to be the main motivation of thousands of biologists and physicians, sport scientists, and mostly students. Thank you very much. I appreciate your time, your knowledge, your science, and the most important thing, your passion.
Wow. Thank you. You have a big head.